Hey, welcome back to this lecture on mutations. In the last lecture, what I went over with you guys was what happens when we have a change in a DNA sequence in the coding region of a gene. The part that actually gets transcribed into mRNA, and then most of the time that RNA is going to get turned into a protein through a process called translation. Or I should say more accurately that RNA will get read and then amino acids will form to form a protein in a process called translation. We talked about what those mutations are in that coding region, but what's interesting about DNA is lots of times, especially in high school and really even undergrad and college, what we talk about a lot of the time is protein synthesis and just the coding part of genes. We forget that there's lots of other DNA that's involved in the expression of genes. Two of those regions are enhancers, which you see here in blue, and then there's also promoters, which you see here in yellow. The first thing that I'm going to talk about today are enhancers and what happens specifically. What are enhancers? One, but then also what happens when we mutate something in an enhancer. Again, these are just sequences that are a little bit upstream of a coding part of a gene. Okay, so the analogy that I'll make for an enhancer is let's pretend that the coding part of a gene is like a light bulb, like a little teeny tiny light bulb. An enhancer is almost like a timing switch in a specific area. It will switch on at certain points in development, or it can also tell us about place where a protein might need to be expressed. So let's look at some examples of that before we get to this other part called a promoter. So in enhancer mutations, what they can actually do is affect the expression of a gene and the expression of an RNA. Right? Then that can actually have multiple enhancers that control timing and location or even the amount of expression of a gene. So let's look at this example here, and let's say that we have this small embryo of an animal, right? And gene A, shown here in orange on this little chromosome, which has a few parts to it, it needs to have that mRNA that's going to be expressed from gene A. It needs to have that present for its development of both brain and limbs, right? So we need to have those things switched on in those cells. So that's why we have a brain-specific enhancer, some A's and C's and T's and G's, and some limb-specific enhancer, some A's, C's, T's, and G's. Now, if we want this RNA to get made in brains, this enhancer needs to be active. If we want this to be made in limbs, we need this to be active. So if we run an experiment like scientists have done and they say, well, let's knock out let's delete this portion of this chromosome for the brain-specific enhancer, what would you expect to see? Well, what I would expect to see is this result. Only the limbs are actually now going to develop. But because I didn't have this signal that said, hey, this needs to be made in brain cells, that mRNA is not going to get expressed. So we'll only see that in the limbs if we go and try to find it. Now, let's say that we have the opposite scenario. And in this opposite scenario, we're going to block out the limb-specific enhancer, and we'll have the normal brain-specific enhancer still switched on. Well, in that case, gene A is only going to be expressed in the brain and not in the limbs. So this is a good example of an experiment that would be run by scientists to determine where an enhancer is and how it's controlling a specific gene. And we could trace the mRNA from that gene and figure out where it goes inside of an organism, right? So enhancers, what they're really great for is that they control the timing, the location, or even the expression, the amount of the expression of a gene. And in this case, what we're looking for really is the location, right? Because we were looking at limbs and brains, okay? Here's another example, and this is another really cool real-life example where a mutation in an enhancer or a change can change the timing or the place where this thing is expressed. So this is our good example in embryonic development. The formation of ribs is controlled by the expression of really a few genes. So these genes are over here, and they're called Hox genes. So in a wild-type mouse, what we're seeing here is 3 different versions of Hox genes called Hox4, which controls development of the neck, Hox6, which controls development of the ribs, and then Hox10, which controls development of the lumbar region or the spine. So in a wild-type mouse, all of these Hox genes, these enhancers, will be turned on, and they'll show you a specific amount and in a location, right? Now, what if we played around with that and what we tried to do was turn up the Hox genes or turn down the Hox genes? So here's a really cool example. This is what a mouse would look like if the HOX6 gene was experimentally repressed. And repressed is just a fancy term for shut down or turned off, right? So we're seeing that this green is representing HOX6 in this chromosome is not present in this chromosome. So the phenotypic expression you can see of that is that there isn't any ribs here. You've got this really strange. You've got this really strange looking animal now, right? Because there's no rib formation at all. Now, what would happen, do you think, if we actually expanded the HOX6 gene? What if we just turn that switch up to 11? What would you expect to see in this mouse? Probably something like that, right? And this is a real image. So this is the HOX6 gene actually expanded, which is going to form tons of ribs now phenotypically compared to the wild type mouse because we've expanded that enhancer. And finally, is there anything that you know of that has like this long slithery body with a lot of ribs in it? This is an image of a snake, right? And what they have is we can actually look and see their HOX6 gene is turned all the way up, right? They have lots of it. So they form lots of ribs and have this long, slender body with lots of ribs and a lumbar region. And that's actually been evolutionarily selected for. So not in the lab, but out in the wild has selected for these genes. So those are examples of enhancers. Now, our last region that we're going to talk about are called promoters. And these are sort of the dimmer switches of the gene world. They're going to control how tightly RNA polymerase is actually going to bind to some DNA initially. It's going to recruit RNA polymerase. And some of them are really strong, which means they make lots of product from the gene, from the coding part. Or they can be really weak and that RNA polymerase won't make as much. So they control the amount of a gene product that's actually made. One example of how this works in the body is actually in diabetes. So normally in the pancreas, this little protein called insulin is made and secreted. And then that protein called insulin is going to go into our blood system, and it's going to regulate the amount of glucose that we have in the blood, which is a really great thing for after we start digesting foods so that we can regulate the uptake of glucose. Without insulin, we can get really high glucose levels, and that can be bad, right? So what we can actually trace this to, and what scientists have done, is you can look and you can see there's mutations in promoter regions which turns down. Remember, like the analogy that I'm using here is it's like a dimmer switch. So instead of turning things up really brightly, it turns things down because of mutation, excuse me, that you see in their promoter regions. What that leads to is if people inherit these types of mutations is it can lead to type 1 diabetes where not enough insulin is actually produced by the pancreas. And that's because of mutations that we can see in the promoter region. So let's rewind and sort of look at what are the big take-home messages here. And then I want to expand on one more point with you. The enhancer regions, right, don't have any impact at all on an amino acid sequence. They don't have any impact on the protein because they're not involved in the coding part. However, mutations in enhancer regions can increase or decrease the amount of products, or it can cause the production in the wrong place or the wrong time, right? And that could be through substitutions or it could be through indels. We also have mutations that can occur in these promoter regions, which are like the dimmer switches. And again, no amino acid is actually going to be impacted here, right? We can't have silence, missense, or nonsense mutations. We can't because there's no amino acid impact. However, there can be impacts on the production of that gene, right? It can increase or decrease in amount due to our certain types of mutation types. Okay, so these are going to control expression, and changes here can actually change the protein, and that's an important distinction, I think, to make. I also want to talk about these last 2 points with you. So we just got done talking about what part of the gene is mutated. Let's talk about the function of the affected gene. And this is going to get us into why we're doing the PCR that we're doing, hopefully. It will give you some background. In physiology, or in trying to maintain normal functioning inside of our bodies, there's genes that will control body function. And when those genes go awry, it can lead to a host of diseases. I'm just showing you some here that are some examples, right? One that we've talked about ad nauseum in class, and we've talked about all these in class, but this is the one that we're going to focus on here because it's the least serious. lactose tolerance. What we can see here, and here's the structure of this structure. Sorry, guys, man, I must be a little tired. Here's the structure of the lactase gene. Remember, lactase is that little enzyme that breaks down lactose. There's a promoter region here where the RNA polymerase is going to go, the dimmer switch, and then there's an enhancer. And what we've seen through studies is that if there's a change, a mutation in the enhancer region for the lactase gene, if we change that from a C to a T, like a substitution, it correlates with lactose tolerance, people actually being able to drink milk well into adulthood, which is actually the mutant version. Lactose intolerance is sort of the wild type or normal version. So what we're going to do is we're going to run a PCR on this region to see whether you have a C or a T in this spot, or if we have any other mutations in that spot that could actually cause the lactase gene to turn off over time, right? The other way we could look at this is genes whose function is to contribute to the formation of organs or specific cells, like we looked at with those Hox genes. You could also look at it with webbing in different birds, right? Like whether or not these genes are turned on in these areas to form webbing. So in the embryo of a chicken and a duck, you can see that there is this webbing, right? But as an adult, the duck will maintain it and the adult will not, right? So some of these genes actually get turned off over time for development. We can also see it and study it in flies, where we can have a normal fly here with just 2 wings, and here we have a total of 4 if those genes are turned on. It's also really important to keep in mind when we're talking about mutations that they're not always lethal. Nor do they have really profound effects or changes on organisms. You know, sometimes there can be mutations, but there aren't these big changes that necessarily occur. And this is an example in a biological favorite, a model organism called fruit flies. And these fruit flies, their fancy name is Drosophila melanogaster. And in these little guys, they have an 8 base pair insertion, or what we'd call a frame shift mutation. Remember, it's an indel, an insertion or a deletion. In the B gene, and that results in the B1 allele, and you can see what the changes are in this B1. It results in just a darkened crown of pigment on their backs. That's it, right? Like so there really is no effect on viability or their survival or their behavior, really anything. So it's kind of a neutral mutation, but that mutation is around. Right? So they're not always these lethal, really bad things, but what we're interested right now in biotech is like, how do they relate to diseases, specifically in humans, right? Finally, what I want to talk about is the timing and place, or the cell type of a mutation. Let's pretend here that in this animation, what we're showing is we've got an egg here and a sperm cell here. And what we've got is we'd call these formations would form somatic cells or body cells, not the sperm or the egg cells, right? So what these are going to do is pass along their genetic information, right? And then what we'll eventually get over a long period of time is the same type of frog, and there might eventually here be a change, right? And what this change here is representing is actually a change in a body cell, right? So while the organism is already formed, right? And this is going to affect the individual in whose body that mutation occurs, but these will not be passed along to the offspring. In order for a mutation to be passed along to the offspring, a different type of change has to occur. And that's what we're going to show here. A germline or a sex cell mutation needs to occur, right? They're not going to affect the individual. That's not going to change their phenotype. But a change in a sex cell, like we're showing here, can be passed along. And that's an important distinction to make. So in somatic mutations or body cell mutations, it affects the person whom the mutation occurred, but they're not passed on to future generations. And cancers are a really good example of that, most cancers. In germline mutations, it does not affect the person where the mutation occurred, but is passed along to all future generations. And that's an important distinction to make. For things to be passed along, they happen in sex cells. For things to affect the actual organism, they happen in somatic cells, and cancers are good examples of that. Germline mutations, some good examples are actually like genetic conditions in humans.